For my biology role model project, I chose Dr. John Craig Venter. Right after graduating high school, Dr. Venter joined the Medical Naval Corps, where he is a veteran for the U.S. Navy. He then got his BA in biochemistry and later on his doctorate in physiology and pharmacology at the University of California, San Diego. After that, he moved to Buffalo, New York, where he worked at the Neurochemistry Research Department at the State University of New York. After that, he went on to work at the National Institute of Health, where he began studying genes involved in signal transmission between neurons. Where Dr. Venter's involvement in science stemmed from was being frustrated with traditional methods of gene identification, which he found to be slow and time consuming. In his free time, Dr. Venter enjoys swimming, surfing, taking helicopter rides, spending time with pets and family, taking nature walks, as well as sailing. All of this can be found on his Instagram, as well as that photo in the upper corner where he is on a helicopter ride with a loved one. What I really admire about Dr. Venter is his development in new ways of approaching synthetic biology. I believe that the way that he tackled the questions that he had about the methods in science that had to do with genomics was really admirable, how he really tackled them head on and with confidence and determination. He pioneered the way for new synthetic biology research, and I really appreciate how he didn't just sit around for someone else to do it. I think that's where I really can relate to him, is that if I have questions about science or biology in general, I don't like to let them fester or sit around. I really like to tackle them head on and see where improvements or changes can be made. I also really admire how he created a nonprofit institution that advances in genomic research in the environmental field, in the synthetic biology field, as well as just improving overall human health. Where Dr. Venter specializes in the field of biology is genomic research. He tackled the question of how to identify genes quicker and better using expressed sequence tags. And then later on, when he created the John Craig Venter Institute, he tackled how to sequence the human genome using cellular genomics. And then, as mentioned earlier, he did dabble a bit in neurochemistry research. The primary literature that I found and used for Dr. Venter is an article where uh, it discussed the xenotransplantation of um, a pig heart into a non-human primate, and this non-human primate in specific in this experiment was a baboon recipient. As you can see in the data figure, the top column is the normal heart as well as normal heart tissue, and then intestinal hemorrhage that was identified in the pig. And then when um, the heart was transplanted into the baboon recipient, it was rejected, and formation developed in the heart tissue, and then cyto loss and fibrosis. And basically, just the conclusion and themes of this experiment was that the pig heart was rejected by the baboon recipient, and it caused a lot of complications. Another primary literature that I found was a book that was written by Dr. Venter as well as David Ewing Duncan, titled The Voyage of Sorcerer, the second edition, I'm assuming, and it dives pretty much just into the microbiome of the ocean. So far, Dr. Venter and his team have found how to sequence human genomes at a faster pace. And what's next for him is new and innovative ways of continuing to sequence and identify not just human genomes, but pretty much all organismal genomes. Overall, Dr. Venter has created new and innovative ways of tackling genomic research. All of his accomplishments are very admirable, and the way that he tackled his questions head on pioneered and just created new paths for a ton of different questions to be answered in synthetic biology. So, thank you for listening.